This week's lecture material is based on information from Chapter 27 in your textbook, Fluid, Electrolyte, and Acid-Base Balance in the Body. Let's review the definitions of intracellular space and extracellular space. Intracellular space refers to that area inside of the cell, or the space inside of the cell, and the extracellular space is that area outside of the cell, or between cells. So if we were to add up all of the intracellular space, of each cell in the body, of the billions of cells in the body, we add up all of those intracellular spaces, that gives us the intracellular compartment of the body. If we were to add up all of the extracellular space, all of the space outside of the cells and between cells in the body, then that's known as the extracellular compartment. Intracellular fluid, then, is the fluid found inside of cells in the intracellular compartment. Extracellular fluid, on the other hand, will be the fluid found outside of cells and in between cells. And when we're talking about blood, the blood within blood vessels, the extracellular fluid that's found outside of blood cells in blood is known as plasma. And everywhere else in the body, the fluid found outside of cells and in between cells in the tissues is known as interstitial fluid. This chart or this image will show you the distribution of water throughout the body between the two major compartments, so the intracellular compartment and the extracellular compartment. We know that in healthy males, approximately 60% of body weight is due to water. In females, the number is a little bit lower. Approximately 50% of body weight is due to water. And the difference being that you find skeletal muscle has a lot more water as opposed to adipose tissue or fat tissue. And generally, females have a higher fat percentage in their body content. As well, they have less skeletal muscle. So that reflects the difference in the percentage of total body weight that's due to water. You'll see that the intracellular fluid volume is approximately 2/3 of the water in the body is in intracellular compartment, and so the other 1/3 will be in the extracellular compartment. And remember that the intracellular fluid or the intracellular compartment refers to all of that fluid inside all of the cells of the body, whereas the extracellular fluid is the fluid found outside of cells and in between cells. And recall that the fluid in blood, in the blood vessels, outside of the blood cells is known as plasma. And the interstitial fluid then is the fluid found outside of cells throughout all other tissues in the body. Water is known as the universal solvent, and this is what this refers to is the fact that many substances, particularly polar substances, molecules that have charges associated with them, can easily move within water or be dissolved in water. So this has to do with the structure of the water molecule, H2O. We see that the oxygen atom has a slight negative charge and the hydrogen atoms have slight positive charges. So if we take a molecule of sodium chloride, which is salt, we see that the positive charge on the sodium ion is attracted to the negative charge of the oxygen atom, and the negative charge on the chlorine atom ion is attracted to the positive charges of the hydrogen atom. So this will dissociate or break apart the sodium chloride molecule and basically dissolve within water. So in this case, we know that water is the solvent. It's the fluid that we're immersing the sodium chloride in, and the solute then would be sodium chloride. But anything that is able to interact or to be put into a solvent like water is known as the solute. We look at the different categories of solutes that we find in the body. We have two broad categories, electrolytes and non-electrolytes. Electrolytes generally refer to substances that are potentially could create an electric current, so there tends to be significant charges associated with these structures, positive and negative charges. Examples being sodium ions, potassium ions, chlorine ions. Salts, bases, some proteins that we find in the body are all electrolytes. Non-electrolytes then are structures like glucose, fats, lipids, creatinine, and urea, which are byproducts of cell metabolism that for the most part don't have charges, significant charges associated with them. So in effect could not really be used if we wanted to create an electrical current. We can't use non-electrolytes to do that. So this is a very important point. Electrolytes, structures that have charges, positive, negative charges associated with it. When we have these electrolytes in association with water, electrolytes have a very powerful effect in being able to move water. So when we want to shift water fluids between compartments, between intracellular and extracellular compartments in the body, we can move electrolytes first to create that movement of water. So for example, if we wanted to first move sodium, a positively charged ion, across a cell membrane, we know that the positive charge of the sodium ion will then attract the negative charge of the chlorine atom, so the chlorine will tend to follow sodium when you cross a membrane, and then we know that water will want to follow the sodium chloride now. So sodium and chlorine together make up salt. We know that water is attracted to salt. So these electrolytes have the ability to move water across cell membranes from one compartment to another. So electrolytes generally have a much stronger effect than non-electrolytes in moving water from one compartment to another. We know that the intracellular and extracellular fluid compartments have different concentrations of electrolytes. Positively charged electrolytes are known as cations, and negatively charged electrolytes are known as anions. So if we look at the intracellular space, the intracellular compartment, the chief cation is potassium. We have much more potassium on the inside of the cell than we do on the outside of the cell. And the chief anion in the intracellular space is phosphate. Phosphate is a negatively charged structure, a negative electrolyte. If we look at the extracellular space or the extracellular compartment, the chief cation is sodium. We have much more sodium on the outside of the cell than on the inside. And the chief anion is chlorine. We have much more chlorine outside of the cell than we do on the inside of the cell. Remember that in the cell membrane that we have sodium and potassium leaky channels. So both sodium and potassium will want to diffuse across the cell membrane down their concentration gradient. And if allowed to continue, this would continue until the sodium and potassium would have equal concentrations on both sides of the membrane. And that's not what we want, because we want to create a negative resting membrane potential. The inside of the cell membrane needs to be negatively charged at rest, and this was what creates this the ability of cells to create a action potential or a nerve impulse. So remember that we have to have these sodium-potassium pumps in the cell membrane that will continually readjust that concentration gradient of sodium-potassium, so we always have more sodium on the outside than inside, and more potassium on the inside of the cell than on the outside. This figure is in your textbook, and again it's just illustrating the concentration differences of the electrolytes in the intracellular and the extracellular compartments in the body. So remember that blood plasma, represented by the red bar, and interstitial fluid, represented by the blue bar, are both components of the extracellular compartment or extracellular fluid, and then the tan colored or light colored bar will be representing the intracellular fluid. So again, you see that in the extracellular compartment, sodium has a high concentration as opposed to the intracellular compartment, and in the intracellular fluid we have much more potassium than in the extracellular fluid. And again, chlorine much higher in the extracellular fluid than on the intracellular space, and phosphate, which is HPO4 2 negative, much more on the inside of the cell than on the outside of the cell. On average, human beings lose about 2.5 liters of water a day, and so water that's lost has to be replaced. So to remain properly hydrated, water intake must equal water output. So where we get water from, we're going to get approximately 1.5 liters of that 2.5 liters will come from the fluids that we drink. 30% will come from the food, water within the food that we consume, and about 10% will come from the water of metabolism. So this is as you recall, in the electron transport chain, the final acceptor of the high energy electrons is oxygen, and that the oxygen combines with protons, hydrogen ions, to make water. So that's the water of metabolism. And then we lose the 2.5 liters a day that we lose, so about 60% will be in the form of urine. And then we have what's known as insensible losses. So this is fluid loss that's not obvious. For example, sweat is, we normally can see sweat, so about 8% of fluid loss is due to sweat, but other types of losses that can occur through the skin that will cause basically an evaporation of the fluid, so it doesn't look like visible sweat, but we're still losing that fluid. So these are known as insensible losses. Same as when we breathe out, the air that we breathe out of our body when we exhale is humidified. So it's not moisture that you see, but we're still losing moisture. So these are insensible losses through the skin and the lungs. And then finally, also in our feces, when we have a bowel movement, we're also losing water in our feces as well. So we have to have a proper balance to remain properly hydrated. The water that we lose each day has to be replaced by intake. If water loss is greater than gain through the day, then we start to become dehydrated. And we know that if we're becoming dehydrated, that we will develop a sensation of thirst, that we want to consume fluids. There's an area in the brain that's called the hypothalamus, and the hypothalamus is able to monitor plasma osmolarity. So this is dealing with the concentration of solute in blood plasma, so the fluid or the extracellular fluid of blood. If you're becoming dehydrated, then the amount of water in blood is becoming less. So in effect, the solute, all the different substances, proteins, cations, anions that are in blood, that concentration of that stuff will start to increase as you're losing, or the relative concentration as you're losing water. So the blood itself actually becomes more sticky, or what's known as more increased viscosity. So this is also reflected in what's called plasma osmolarity. So the more dehydrated you become, the less water in the plasma, which means that there is relatively more stuff in the plasma. And the hypothalamus can monitor this, and if plasma osmolarity is increasing due to dehydration, it will stimulate the thirst center that will make you want to consume fluids. So the major stimulus then for stimulating the thirst center in the brain is plasma osmolality. When plasma osmolality increases due to dehydration, the osmoreceptors in the hypothalamus will fire, and then that will tell the thirst center that's in the hypothalamus to create a sensation of thirst, so that it will make you want to drink water, fluids, something to try to satiate that thirst. And then as we're consuming these fluids, we're going to have other receptors in the mouth and the throat that are going to respond to that moisture, as well as stretch receptors in the stomach as water is entering, and in the intestine will stimulate or start to satiate that sensation of thirst, make you feel less thirsty. And then as water is being absorbed out of the GI tract and back into your blood, the plasma osmolality will begin to decrease, and now the osmoreceptors are going to stop firing in the hypothalamus, and that's going to take away that sensation of thirst. Another mechanism is stimulated as you become dehydrated, and this is related to the release of antidiuretic hormone from the pituitary gland. So the antidiuretic hormone is made by the hypothalamus, but it's stored in the pituitary gland. Pituitary is a structure in the brain as well. And when plasma osmolality is increasing due to dehydration, your pituitary will release antidiuretic hormone. Antidiuretic hormone acts on the kidneys to create a more concentrated urine, which means it's going to not allow as much water to be lost in the urine. And we know that if you drink several drinks of alcohol, for example, you start to notice that your volume of your urine starts to increase, and also it becomes more dilute. So there's less color. The old saying, drink beer, pee clear. And that's because alcohol inhibits the release of antidiuretic hormone from the pituitary. So it results in you creating a very dilute urine. You're losing water, more water in your urine. The name antidiuretic, diuresis refers to the act of urination. So if you take a diuretic, for example, diuretics are a class of medications that people take for blood pressure, trying to reduce their blood pressure. Diuretics make you pee more so that you lose more volume of water in your urine, which will decrease your blood volume, which will help to decrease blood pressure. Now, so if you have low levels of antidiuretic hormone that's being released, so in the case where if you're very hydrated, if you've drank a lot of water, we want to get rid of some of that water. So in this case, you're going to have decreasing plasma osmolality because you're very hydrated, your blood is very hydrated. In that case, an inhibition of release of antidiuretic hormone will result in the kidneys making a more dilute urine. So again, this action is being monitored by the osmoreceptors in the hypothalamus. This slide, again, will emphasize the actions of antidiuretic hormone. Antidiuretic hormone is also known as vasopressin. So if you see that term vasopressin, it's referring to antidiuretic hormone. So if we look at, first we talked about increasing osmolality in the blood, so due to dehydration. This can also be correlated with an increasing sodium concentration in the plasma. So if you have a lot of sodium in your plasma, then it's going to effectively cause an increase in osmolality. This will stimulate osmoreceptors in the hypothalamus. The hypothalamus then will tell the posterior pituitary to squirt out antidiuretic hormone, which goes into your circulatory system. And then the ADH, antidiuretic hormone, will act on the kidneys. That's the target of the antidiuretic hormone. And it'll cause the kidneys to increase water reabsorption. So to take water out of the urine filtrate that's being made and put that water back into the circulatory system, back into your blood vessels. And as this happens, this is going to result in a scant or a concentrated urine. And also the blood osmolality will start to decrease and the plasma volume will increase. And as plasma volume increases and blood osmolality decreases, this is going to tell then the osmoreceptors in the hypothalamus to stop releasing antidiuretic hormone. So that's known as a negative feedback inhibition that occurs. So this is how we monitor or create a homeostatic level of osmolality and blood volume that we require in the body. If you look on the right side of this flow chart as well, we see there's another stimulus, decreasing plasma volume or decreasing blood pressure. Will also cause a release of antidiuretic hormone. In this case, with decreasing blood pressure and plasma volume, we have stretch receptors in the walls of a couple of the major arteries in our body. And these are called baroreceptors. And if these baroreceptors are not being stretched enough because the blood volume is decreased or blood pressure is too low, that will stimulate the posterior pituitary as well to release antidiuretic hormone. And the overall effect of that then is to increase blood volume, increase plasma volume, which will also result in an increase in blood pressure. Because remember, we need to have enough blood pressure as well to be able to perfuse organs and tissues in the body. If blood pressure becomes too low, organ failure can start to occur where we're not getting enough oxygen and nutrients to those tissues. A major premise in the body is the movement of water in relation to sodium and chloride. So generally throughout the body, when we're trying to move water from one compartment to another, first we will move sodium. That tends to be the primary cation or electrolyte that is moved, and then sodium is a positively charged ion. We know that chlorine, negatively charged, will follow sodium, and then we have salt, sodium, and chloride together. It has a very strong attraction for water. So if we want to move water across a cell membrane from one compartment to the next, we can actively transport sodium first. Chlorine will follow the sodium across the membrane, and then water will follow the sodium and chloride. So extent of urinary salt loss is the main factor that determines body fluid volume. If we want to increase the amount of body fluid that we lose through urine, we can add more sodium chloride into the urine filtrate, and water will follow sodium chloride, which means that will be excreted in the urine, and we can reduce the amount of fluid or water in our body fluids or body compartments. The main factor that determines body fluid osmolarity then is how much water we lose in the urine. As we lose more water in the urine, our plasma osmolarity or osmolality will increase. We have three hormones in the body that will help to regulate the renal or kidney sodium and chloride reabsorption. Angiotensin 2 and aldosterone, these two hormones will promote the kidneys to reabsorb sodium and chloride out of the urine filtrate and back into the blood. So when we move sodium and chlorine out of the urine filtrate back into the blood, water will follow sodium and chloride by osmosis, and this is a mechanism that occurs when we're becoming dehydrated. The other third hormone is called atrial natriuretic peptide, ANP. The word, first of all, atrial refers to a chamber in the heart, the atrium, and there are cells in the wall of the atrium that produce this hormone. Now, natriuretic, the word natriuretic, natri, N-A-T-R-I, refers to sodium, and this is where we get Na for sodium. The chemical symbol for sodium is Na, comes from natri, which I believe is either Greek or Latin for sodium. So this is a hormone that's produced by the atrium, a chamber in the heart, and these cells are able to respond to increased stretch in the wall of the atrium. So if there's an increased blood pressure, blood volume, if there's a high volume of blood coming back into the heart, that causes a stretch in the walls of the atrium. And the atrium will release ANP, atrial natriuretic peptide, and this again acts on the kidney, and it results in the kidney excreting more sodium and chlorine into the urine filtrate, and again water will follow the sodium and chloride and results in an increased volume of urine along with the sodium and chlorine, and ultimately results in a decreasing blood volume. So now that's going to cause less stretch in the atrium, in the chamber of the heart, which will then stop the atrial cells from secreting atrial natriuretic peptide. This slide will illustrate the effects of the renin-angiotensin aldosterone mechanism. So if we look at the top of this flow chart, we see that the stimulus is going to be either decreasing sodium ion concentration or an increase in potassium concentration in the blood. And these two ions are normally act opposite to each other because they tend to go in the opposite direction across the cell membrane. So If you've got more sodium going one way across the membrane, you tend to have more potassium going the opposite way. And this has to do with the sodium-potassium channels, pumps that we know about. So either low sodium or high potassium concentration in the blood will stimulate the renin-angiotensin mechanism that starts in the kidney. Angiotensin, we'll do this in more detail when we look at the kidney physiology in the second half of the semester. But once we've made angiotensin, angiotensin will act on the adrenal cortex. The adrenal glands sit on top of the kidneys, left and right kidneys. We have the adrenal glands. And the angiotensin will stimulate the adrenal cortex, the bark of the adrenal gland, to secrete aldosterone. Aldosterone is a hormone that's going to act on the kidney. So the kidney tubules it'll result in them reabsorbing more sodium. So you're going to pee less sodium. You're going to pull more sodium out of the urine filtrate back into the blood. And conversely, as sodium is going into the blood, means that more potassium is going into the urine filtrate. So you'll tend to urinate more potassium out. And as a result, eventually you restore the homeostatic plasma levels of both sodium and potassium. These ions, sodium, potassium, and particularly potassium, has a very narrow range of homeostatic levels. You can't have too much or too little potassium. There's a very narrow range that is normal. Otherwise, you can start to create very serious problems, especially dealing with the heart and atrial fibrillations and arrhythmias that can occur due to potassium levels being too high or too low. So sodium is the major electrolyte, the major cation that is responsible for the movement of other electrolytes and water in the body. So as a basic premise, when we want to move fluid or water from one compartment to another, or we want to move electrolytes from one compartment to another, we're going to move sodium first. Well, we can actively transport sodium across the cell membrane, and other electrolytes will follow either in the same direction or opposite direction, and as well water tends to follow sodium. So if we look at the hormonal regulation of sodium and chloride in the body, we start at the top here. We see that if we have an increased intake of sodium and chlorine, so if we're consuming salt, that's going to increase the blood concentration, plasma concentration of sodium and chloride ions. And by osmosis, water is going to be attracted to that sodium and chlorine that's in our blood. So from the interstitial fluid, the fluid in the tissues will be drawn into the blood vessels towards the increased concentration of sodium and chloride. And this is going to result in increasing blood volume. So as blood volume increases, it's going to cause a stretch in the atria, the walls of the atria and the heart. These cells in the atria then will release atrial natriuretic peptide, and this is a hormone that will act on the kidneys to reduce the amount of sodium being recovered from the urine filtrate. So this means that more sodium stays in your urine. Water will be attracted to the sodium as well as chlorine, and so which means that you will pee out more sodium and chloride, and you will pee out more water. And the overall effect of this is that you will have a decrease in the volume of water in your blood, which will decrease blood volume and also decrease blood pressure. As well, we see back up to the middle of that flow chart with increased blood volume. That means more blood is being filtered through the kidneys, which means there will be less of the renin-angiotensin mechanism that will be stimulated, which means less aldosterone will be released by the adrenal cortex. And again, as a result, remember, aldosterone acts to increase recovery of sodium from the urine filtrate. So in effect, it's going to cause you to put more sodium into the urine. And remember, when we move sodium, chloride follows sodium into the urine, and as well water will then follow the sodium and chloride. We're now going to look at acid-base balance within the body and how the body is able to maintain homeostatic levels of pH of the body fluids. This is a major homeostatic challenge because most metabolic reactions that occur in the body create acids. So with acid formation, we're creating increased acidity of fluids, and tissues, cells are very sensitive to pH levels or the acidity of the fluids themselves. So it's a challenge to be constantly trying to maintain a very narrow range of pH and trying to neutralize these acids that are being produced. When we look at proteins, for example, we know that proteins are essential for all functions that occur in cells and in the body. And remember that the three-dimensional shape of the protein that's created in protein synthesis that we learned about associated with the ribosome, that three-dimensional shape really is important for the functioning of the protein. And with increasing acidity, these three-dimensional shapes are very sensitive to the pH of the fluids that they're in, and if fluids are becoming too acidic, they can actually start to damage or rearrange that three-dimensional shape, and the proteins become inactivated. We know generally that diets that have large amounts of proteins, so if you consume a lot of meat in your diet, you tend to create much more acids as opposed to bases, which tends to create an acidified blood. Normal pH of body fluids is between 7.35 and 7.45. So you do have to know that number. Remember that the pH scale goes from 0 to 14. 7 is neutral. Everything below 7 is acidic, and everything above 7 is basic. Another word for being basic or having more base than acid is alkalosis or alkalemia. So emia refers to blood. Whenever you see that suffix emia, it's referring to blood. So alkalemia refers to increased alkalinity of the blood. So arterial blood that is going to have a pH above 7.45 then, because remember homeostatic level is 7.35 to 7.45. So if your pH is above 7.45, it means that you're becoming alkaline, that you have more base than acidic, more base than acid in your blood. So this is known as alkalosis or alkalemia. On the other hand, if you have a pH that is below, arterial blood is pH is below 7.35, this is known as acidosis or acidemia. So even though 7.35 is above neutral, remember in body fluids the normal pH is 7.35 to 7.45. So if you drop below 7.35, you're becoming more acidic. So this is known as acidosis or acidemia if your pH is below 7.35. And recall that pH is determined by hydrogen ion concentration. So the term pH refers to the partial pressure of hydrogen ions or the concentration of hydrogen ions. So the more hydrogen ions you have in your fluids, the more acidic you are, the lower your pH is. So most of the hydrogen ions that are developed in the body originate from cellular metabolic reactions. For example, breaking down proteins will result in the release of phosphoric acid into the extracellular fluid. The anaerobic respiration of glucose that we learned about in the cytoplasm of the cell can produce lactic acid. Using fat as a source of fuel, so fat metabolism will result in the production of ketone bodies known as ketoacidosis. And transporting carbon dioxide in blood as bicarbonate ion releases hydrogen ions, and we'll see that in more detail when we look at blood and respiration. But all of these are normal metabolic reactions that occur in the cells of the body, and they all result in the production of hydrogen ions or acidity. So they contribute to the acidity of body fluids, and we have to have systems available in the body to neutralize these acids, otherwise we'll become too acidic. We basically have three lines of defense when we're trying to neutralize the acids that are being built due to cellular metabolism. The first line of defense would be the chemical buffer systems that are in the blood, and these act very quickly within seconds to try to neutralize these acids that are being built due to cellular metabolism. Secondly, the respiratory center in the brainstem will determine how quickly we breathe and what our depth of respiration is. And this is important because carbon dioxide, which is carried in our blood, remember carbon dioxide is a waste product of cellular metabolism, carbon dioxide is directly correlated with acidity. So the more CO2, carbon dioxide, you have in your blood, the more acidic your blood is. So if we increase our rate of respiration, we can blow off more CO2 to try to lower our acidity in the blood. So this occurs within 1 to 3 minutes as a second line of defense. And then the third line of defense will be the kidneys. The kidneys are very powerful mechanisms or have very powerful mechanisms in trying to normalize pH levels in the blood. But these mechanisms normally take a little bit longer to kick in. They could take hours to days before these renal mechanisms have their effect in neutralizing the acids. We'll look at the chemical buffer systems first that are present in blood. And these systems act very quickly. Remember, they're the first line of defense, and what they'll do is act by binding to these hydrogen ions to try to neutralize the acidity. So they don't actually get rid of the hydrogen ions from the body or the acids from the body, and that's when we look at renal or kidney mechanisms. We'll see that the kidneys can actually excrete acids or hydrogen ions, so you're actually peeing out acids to try to make yourself less acidic. But these chemical buffer systems will just bind hydrogen ions or the acids to neutralize them. So three major chemical buffer systems that we have in blood is the bicarbonate buffer system, the phosphate buffer system, and then the protein buffer system, and we'll look at each of these individually. So the most abundant chemical buffer system is the protein buffer system that we see in the intracellular compartment fluids and in the blood plasma. So in red blood cells, for example, hemoglobin, which we'll learn more about later in the course, hemoglobin is the protein that binds oxygen in the red blood cell, but hemoglobin is a protein that will also act as a buffer. And as well, in the blood plasma, we have one of the proteins that we have is called albumin, and it's also acts as a major buffer system for the acid-base balance in blood. So if we were to look at the bottom of this slide, a basic idea of a protein structure, we have a carbon backbone of some sort, and these can be very, very large carbon structures, but somewhere on the protein structure, you're also going to have a carboxyl group, which is the carbon with 2 oxygen and a hydrogen ion. And then we also have an amine group, which is the NH2 that you see. So if the blood is, we'll talk about the scenario if the blood is too basic, so which means that it's not acidic enough. And so in this case, what we want to do is to try to make the blood more acidic, because remember it's a homeostatic range of 7.35 to 7.45 of pH, and if our pH is above 7.45, it means we're too alkaline or too basic. So we want to add acidity to the blood to get it back to a homeostatic level. So in this case, we can use the hydrogen ions that's associated, or the hydrogen atom that is associated with the carboxyl group. The hydrogen can dissociate from that carboxyl group and become a free hydrogen plus ion or atom, or proton, sorry, that will add to the acidity of the fluid that it's in. So in effect, it's going to help to decrease the pH to get you back towards a homeostatic range. On the other hand, if your blood is too acidic, it means that there's too much free hydrogen ions, H positive ions. So what we can do now with the proteins is that the NH2 part of the protein can bind another hydrogen to become NH3, and in doing so, it's neutralizing that hydrogen, taking it out of the fluid, and effectively neutralizing it by binding it to that NH3 protein component of the protein structure. The second component of the chemical buffer system is the carbonic acid bicarbonate buffer system. And this has to do with the respiratory system regulation of acid-base balance. So this is a reaction. The two equations you see at the bottom of this screen are referring to the reactions that occur within the red blood cell. And we'll cover these in a little bit more detail when we do our tutorial this week. So I want to make sure everybody understands this process. We'll talk about it by illustrating it more on the board. So there is if we look at the equation, CO2 plus H2O goes to H2CO3, which then goes to hydrogen ions plus bicarbonate. So we know that CO2 is carbon dioxide is a waste product of cellular metabolism, and when we add water to CO2, we make carbonic acid, which is H2CO3. And then this will dissociate into hydrogen ions and bicarbonate, HCO3 negative. So the way chemical reactions occur, the left side of the equation here would be the reactants that we start with, and the right side of the equation would be the products. So if we start with more reactants, if we had a lot of CO2, we're going to create more products on the right side. So with increasing CO2, we're going to eventually result in creating more hydrogen ions and more bicarbonate. So this is where the relationship is between carbon dioxide and pH. So as we have more CO2, it results in more hydrogen ions being produced, which means it's increasing acidity. So if we look at this reaction will result in hydrogen ions, which are acids, and bicarbonate ions, which are bases. So in effect, we can use those hydrogen ions to increase acidity of fluid if necessary, and we can use those bicarbonate ions, HCO3 negative, to decrease acidity if we require. So the bicarbonate can bind to hydrogen ions to neutralize, or the hydrogen ions can be used alone as a source of increasing acidity. Once again, we're trying to achieve a homeostatic range of 7.35 to 7.45 pH. The bottom reaction, HB, refers to the protein hemoglobin, which is the protein that binds oxygen inside the red blood cell. And we know that with increasing concentration of hydrogen ions in the area of the hemoglobin-oxygen complex, it causes the hemoglobin to release the oxygen, which makes sense, and we'll do more detail on this process again in class. And as a result, the hemoglobin will then bind, hemoglobin's a protein, it will bind the hydrogen ions and take it out of circulation, so to speak, and in effect neutralize the acidity that's created by that hydrogen ion. The final component of the chemical buffer systems in blood is the phosphate buffer system, and this is located mostly in the intracellular fluid. And these phosphate structures will bind hydrogen ions. So in our first example, hydrogen ion H positive is a strong acid, and if we combine it with hydrogen phosphate HPO4 negative, we create a weak acid. So in doing this, going from a strong acid to a weak acid, we are decreasing the acidity of the fluids. On the other hand, if we have a situation where the fluid is too basic, too alkaline, so we have these hydroxyl ions, OH negative, which are strong bases. We can combine it now with the weak acid, H2PO4, and as a result we make water, and we create again HPO4, hydrogen phosphate, which is a weak base. So we've gone from a strong base to a weak base. So in this case we've made the fluids more acidic. So in this way we're trying to again get that homeostatic range of 7.35 to 7.45, whether we have to make it more acidic or more basic to get to that range. So we've seen that chemical buffers can tie up excess acids or bases, but they don't eliminate them from the body. They just neutralize them while they're still in the fluids. The lungs, we'll see, can also decrease acidity by getting rid of carbon dioxide. So by increasing our respiratory rate or our depth of respiration, we can blow off more carbon dioxide, and that in effect will create a less acidic blood. But only the kidneys can rid the body of the metabolic acids, so phosphoric, uric, lactic acids, ketones. All these structures that are made from metabolic reactions in the cells can actually be rid of through the kidneys. And this is important because if we can get rid of these structures, these acids, it eventually results in a condition known as metabolic acidosis, where the entire body all fluids in the body are becoming more and more acidic, and can eventually become life-threatening. So only the kidneys have the power to actually excrete or get rid of these acids from the body. So the ultimate acid-base regulatory organs are the kidneys. The mechanisms that the kidneys have for neutralizing acids, getting rid of acids, is extremely powerful. When we look at the kidneys' abilities to regulate acid-base balance, the most important renal or kidney mechanisms for regulating acid-base balance is dealing with bicarbonate ions. So bicarbonate is HCO3 negative, and if we want to increase the acidity of our fluids in our body, we can get rid of bicarbonate ions. So we can put bicarbonate in the urine to pee it out. So in effect that's going to raise the acidity of the blood. Conversely, if we want to decrease the acidity of the blood, we can reabsorb or take bicarbonate out of the urine filtrate and bring it back into the blood to try to create a less acidic environment within the blood. So in either case, you're manipulating the concentration of bicarbonate ions. Either you're keeping more in the blood or you're getting rid of more in the urine. If we look at this diagram here, this figure, so this is a representation of cells, tissue within the kidney. The peritubular capillary that you see there will be, that's going to be the blood that is part of our systemic circulation. So we see there bicarbonate ions, in brackets nu, HCO3 negative. So if the blood in the peritubular capillary, which represents our circulatory system, if the blood is too acidic there, we can bring more bicarbonate ions, HCO3 negative, into the peritubular capillaries to make that blood less acidic. On the other hand, we can also excrete more hydrogen ions out, and on the left side of that image you see the filtrate in the tubule lumen, so that represents what's going to become urine. And so again, if we're too acidic in the blood, these chemical reactions can result in hydrogen ions being put into the urine filtrate so that we can pee that out in our urine, and in effect cause decreasing acidity of the blood. If we look at the state of respiratory acidosis or respiratory alkalosis, it's important to understand what this refers to. So this has to do with the concentration of carbon dioxide in the blood, and it's going to result from the inability of the respiratory system to get rid of enough CO2 or to balance the pH in the blood by the respiratory rate. So you're going to see problems very commonly in people who have lung disorders, things like bronchitis, emphysema, lung cancer, can result in respiratory acidosis, for example, because they're not able to effectively get rid of as much carbon dioxide as they should be able to because of these pathologies. So respiratory acidosis and alkalosis refers to either increasing acidity or alkalinity of the blood, but due to some process that's occurring in the lungs. So the result is the partial pressure of carbon dioxide, or the amount of carbon dioxide in the blood, is not being adequately regulated by the respiratory system. Okay, so it's important to know that respiratory acidosis is the most common cause of acid-base imbalance in the body. So, for example, if we already mentioned pathologies like bronchitis, pneumonia, emphysema, where there's gas exchange is being impaired in the lungs, it's going to result in rising levels of carbon dioxide in the blood. So this refers directly then to increasing acidity of the blood, or what's known as respiratory acidosis. Respiratory alkalosis happens when the carbon dioxide in the blood is too low. So if you have not enough CO2 in the blood, remember that CO2 correlates with acidity, so if you have very low CO2, it's going to result in your blood being too alkaline. Your pH has risen above 7.45. So this is what you see when somebody is hyperventilating. So if there's a problem with not feeling like you're getting enough oxygen, maybe due to anxiety or even being in a high altitude situation, and so in this case CO2 is too low, so this is when people will breathe into a bag. So when you're breathing into a bag to correct respiratory alkalosis, what you're doing is that you're breathing out carbon dioxide into the bag, but now you're also breathing carbon dioxide back in again. And so in a short period of time, you're raising the carbon dioxide levels in your blood because of this process of breathing in and out of a bag. Metabolic acidosis, on the other hand, has to do with bicarbonate ion concentrations in the blood. So respiratory acidosis is correlated with carbon dioxide levels. Metabolic acidosis has to do with bicarbonate levels. So if the bicarbonate ion, if you don't have enough bicarbonate ion in your blood, it means that your blood is becoming more acidic, which means that the pH is going lower than the 7.35. So causes of metabolic acidosis: either you're losing too much bicarbonate ion, so for example in cases of diarrhea, or if you have a kidney disorder that's causing you to pee out too much bicarbonate ion, or on the other hand, if you have conditions where your body, your cells are making too much acids, so too many acids, so you're producing too much lactic acid, or if you're diabetic, you're breaking down proteins, fats, and creating these acids that are making you metabolically acidic. On the other hand, metabolic alkalosis means that your blood pH is rising above the 7.45 homeostatic level, and this can indicate that there's too much bicarbonate in your blood, so or not enough acid. So for example, if you're vomiting and repeatedly vomiting, and you're vomiting the acid, the stomach contents are very acidic, and if you're vomiting that acid out of your stomach, you're decreasing the amount of acid available to your blood, and so in effect causing the blood to become more alkaline. People who take excessive numbers of antacids, like Tums, those types of things, or in a fluid form that's trying to neutralize the acidity. If people have problems with heartburn, that kind of thing, that if they take excessive amounts of these antacids, it can put you into a state of metabolic alkalosis. And conditions of constipation, where people are not having regular bowel movements, who are and the waste materials staying inside the digestive tube for prolonged periods of time. And it gives your digestive system more time to be able to absorb more of the bicarbonate that's in those digestive juices, and it can potentially cause you to go into a state of metabolic acidosis. It's important to understand that the two systems, the respiratory system and the urinary system, the kidneys, will help to or will attempt to compensate for each other's problems if it's creating an acidosis or an alkalosis state in the body. So, for example, the respiratory system will attempt to correct metabolic acid-base balances by either causing you to hyperventilate or to hypoventilate. So if you're becoming metabolically acidic due to some disorder, your breathing rate tends to increase, and either increase in frequency and in depth of respiration, with the attempt to try to blow off more CO2 and to try to compensate for that acidity, that metabolic acidity, by decreasing the level of carbon dioxide in your blood. Conversely, if you're becoming metabolically alkaline, too basic, your respiratory system will try to compensate by having you hypoventilate, try to slow your respiratory rate down, and make your depth of respiration more shallow, so you're trying to retain more carbon dioxide in your blood to try to compensate for that metabolic alkalosis. And the kidneys will do the same in the response to respiratory acidosis or alkalosis. So if you have respiratory acidosis, too much carbon dioxide in your blood, the kidneys will try to reabsorb more bicarbonate out of the urine filtrate to try to neutralize that acidity.